Hi, welcome to our channel of IGNOU Audiobooks, Indira Gandhi National Open University, School of Sciences, SOS, Bachelor's Degree Programs, Bachelor of Science, Honours, in Biochemistry, BSCBCH. Ability, Skill Enhancement Compulsory Course, AECC. BBCS-183 Tools and Techniques in Biochemistry. Block-1 Tools and Techniques in Biochemistry. Unit-1 Introduction to Biochemistry Laboratory. 1.1 Introduction. Introduction to Biochemistry Laboratory is the first unit of the Skill Enhancement course, Tools and Techniques in Biochemistry, BBCS-183. Biochemistry is a laboratory-based science where biochemists can understand and solve the biological problems by using the knowledge of chemical structure and techniques. A laboratory is a dedicated room that designed to carry out experiment, testing hypotheses and research work under controlled conditions. Although safety and efficiency are the two major concerns in the modern science laboratory, so science student must have complete knowledge of laboratory safety guidelines, physical and chemical properties of chemicals, cleaning and maintenance of glassware. Recall the lab exercise 1. Cleaning and maintenance of glassware, safety measures in laboratories. From the lab course, Molecules of Life, BBCCL-102, where you have learned the laboratory safety rules and cleaning and maintenance of glassware in the lab. Laboratory safety rules ensure us to keep safe and clean laboratory environment. Therefore, in this unit you will learn the good laboratory practice and its significance, laboratory safety measures, general laboratory facility in biochemistry and safety symbol. In next unit 2, you will study biochemical reagents and solutions. Expected learning outcomes. After studying this unit, you should be able to define the good laboratory practice and its importance. Explain basic laboratory facilities and common equipment. Highlight safety practices in laboratories. Enlist the personal protective care equipments, and identify laboratory safety measures and safety symbol. 1.2 Good Laboratory Practice, GLP. Good Laboratory Practice, GLP, deals with the quality management system, which ensure and promote drug safety and quality data for non-clinical laboratory studies across the world. In the early 70s, Food and Drug Administration, FDA, had noticed poor laboratory practices in various toxicology laboratories in the United States, US. FDA found that several testing laboratory conditions were submitted false testing data for approval of pharmaceutical products. This false data had reflected malpractice with inadequate lab facilities. In 1978, Food and Drug Administration, FDA, had created regulatory guidelines to prevent malpractice in the pharmaceutical companies. The GLP regulation was initially published by Food and Drug Administration, US, FDA, for non-clinical laboratory studies and formally accepted and updated in 1981 by the Organization for Economic Cooperation and Development, OECD. These GLP guidelines are also called OECD principles. The OECD is an international organization of 37 countries which involved in making standard guidelines and rules for good laboratory practices. OECD principles for GLP ensure quality management system of reliability, quality test data, safety of chemical products and non-clinical laboratory test facilities. World Health Organization has defined the good laboratory practice, GLP, as a quality management system which is concerned with the organizational process and the conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived and reported. The aim of GLP regulation is to promote and support the efficacy, quality and integrity of test data worldwide. It emphasizes the quality management system for physical, biological and chemical test systems to develop pharmaceutical products. The GLP is not concerned with scientific design but is related to scientific integrity. It helps scientists to obtain results that are reliable, repeatable and worldwide recognition. Therefore, GLP is necessary for management of quality test facility and development of quality test data. Ensure efficacy and safety of pharmaceutical products. Worldwide mutual acceptance of data. Minimize the mistake while non-clinical testing and avoid duplication of data. Reduce technical barriers for marketing and trading and protection of human health and the environmental safety. 1.2.1 Good Laboratory Practice Principles. GLP Guidelines and Principles Concerned to the Quality Management System in research organizations and pharmaceutical companies. GLP guidelines and criteria are meant to manage quality testing system or assessment of the safety, quality and efficacy of pharmaceutical products for human or animals. OECD principles ensures to chemical safety concerned with the environmental health or safety. The GLP regulation also provides enough information in detail about the test, facility management, organization and personnel, quality assurance program, facilities of test system, waste disposal, apparatus, material, and reagents, physical, chemical, biological test systems, receipt, handling, sampling and storage and characterization of the test and reference items. Standard operating procedures, performance of the study, reporting of study results, storage and retention of records and materials. Fig. 1.1. In a nutshell, GLP is a quality system that tells us what type of organization, process, test studies, equipment, test facility, monitoring and documentation are being used for non-clinical testing and environmental health safety. However, India in 2002 has also established the National GLP Compliance Monitoring Authority with compliances of OECD GLP principles. GLP has been mandatory for the pharmaceutical industry, drugs and cosmetics, since November 2010 in India. It is applicable for granting GLP compliance certification to the pharmaceutical industries for marketing of drugs and cosmetics. This applies to all pharmaceuticals products, veterinary drugs, pesticide, cosmetic products, food contamination limits, food products, color additives and medical device H. Look at the screen. Fig. 1.1 Overview of GLPS Principles. A final report is prepared and signed with dated by the laboratory director after completion of each laboratory activity to indicate acceptance of responsibility for the validity of the data in compliances with the principles of good laboratory practice. Therefore, GLP is a set of practices which covers all important aspects related to the organization functions and non-clinical lab studies. Fundamental points of GLP. 1. Test facility organization and personnel. Test facility management's responsibilities. Study director's responsibilities. Principal investigator's responsibilities. Study personnel's responsibilities. 2. Quality assurance program. Responsibilities of the quality assurance personnel. 3. Facilities. Test system facilities. Facilities for handling test and reference items. Archive facilities. Waste disposal. 4. Apparatus, material, and reagents. 5. Test systems. Physical, chemical, biological. 6. Test and reference items. Receipt, handling, sampling and storage. Characterization. 7. Standard operating procedures. Record keeping, reporting, storage, and retrieval. Test system, where appropriate. Quality assurance procedures. 8. Performance of the study. Study plan. Content of the study plan. 9. Reporting of study results. Content of the final report. Information concerning the sponsor and the test facility. Dates, description of materials and test methods. Statement, results and storage. Thus, storage and retention of records and materials. Note, to fully understand the scope of GLP principles, it is probably easiest to start with a look at all of the sections in the given link. https://www.oecd.org/chemical-safety/testing/good yojak chinh laboratory yojak chinh practice glp.htm. 1.3 General Science Laboratory Facility. A laboratory is a designated place for performing experiment as well as test hypotheses. It is the place of new scientific discovery, inventions. Student must know the place, location from where to get chemical and glassware in the laboratory for performing lab experiment. The laboratory glassware, chemicals and equipments play a key role to perform the laboratory experiments. In biochemistry laboratory, the important Laboratory equipments are electronic balance, pH meter, spectrophotometer, centrifuge, light microscope, cooling and benchtop centrifuge, chemical fume hood, incubator, hot air oven, water bath, electrophoresis unit, hot plates, and autoclave, water distillation unit H. These all are permanently placed in a specified area in laboratory as they are frequently used. The uninterpreted electric supply, computer system and washing sink are also essential aspects for laboratory work. A biochemistry laboratory needs a variety of glassware and apparatus, including beakers, conical flask, reagent bottles, test tubes, measuring cylinders, wall mount drying rack, glass pipettes, micropipettes, tips, burettes, thermometer, glass rods, culture vessels, physical or electronic balance, petri dishes, needles, tripod stand, forceps, scalpels, aluminum foil, markers, pens. H. Look at Fig. 1.2 showed some common lab equipments. These include test tube, graduated cylinder, beaker, conical flask, test tube. Laboratory has a common space for storing separately glassware, apparatus, and wide range of chemicals in almirah. These items are provided to students at the time of requirement. Look at the screen. Figure 1.2, Common Lab Equipments. Therefore, Science Laboratory has the following arrangement as shown in Fig. 1.3. Figure 1.3, A view of modern science laboratory. A-B, Laboratory working. Bench with laboratory shelves. C. Glassware cleaning area. D. Store chemicals and liquids in dedicated cabinet. Look at the screen. 1.4 Safety practices in the laboratory. Now discuss the laboratory safety rules which is necessary for all science student while working in the laboratory. However, you might be learned ad. Practiced laboratory safety rules in your 10-2 biology, chemistry laboratory. Science students must learn and practice the laboratory safety rules which protect us from exposure of harmful chemicals and reagents. Biochemistry is a scientific discipline based on experimental work. It requires good laboratory facility to conduct biochemical and molecular experiments. Laboratory is the place where students learn and perform techniques of preparation, identification, and estimation of biochemical substance. However, laboratory is not a safe place as it involves many hazards, chemical, gases as well as toxic reagents. There are many chemicals, gases and reagents are often toxic and lethal, which poses health risk. Laboratory safety guidelines reduce the risk of exposure of chemicals and gases and help in maintaining clean and safe laboratory environment. Laboratory safety rules applies on all laboratory worker, science students, lab assistant, institution, pharmaceutical companies and regulatory agencies. A. Laboratory safety manual should be developed with identification of the major risk area and procedures taken for preventing accidental damage at school, colleges, universities, research institutes and pharmaceutical companies. The laboratory safety guidelines ensure us for achieving a safe and healthy working environment in laboratory. Therefore, laboratory safety rules must be understood and practiced by all science students. A science laboratory can be a very dangerous and harmful if one does not follow the laboratory safety rules. Laboratory workers can expose or contact with the poisonous, harmful chemicals, reagents, and gases H. There may be certain a threat to the health and safety of people at the workplace. These include physical hazard, chemical hazards and biological hazards H. 2. Overcome the laboratory safety issue, you must follow laboratory safety rules. Guidelines before carrying out experiments safely in the lab without endangering yourself or others around you. Carelessness on the part of one student can often cause injury to other students. 1.4.1 Laboratory Safety Rules. General Good Laboratory Safety Practices. 1. Attend laboratory safety training, workshop prior to the start your laboratory work. 2. Follow the instruction given by teacher and perform experiment at allotted place in the laboratory. 3. Read written experimental procedure given in the lab manual while performing laboratory exercise. 4. Read the information and instruction given on the labels on equipment and chemical bottle before use them. Also check warning signs on it. 5. Report damaged electrical equipment to the lab supervisor, teacher. 6. Do not leave active experiments unattended. 7. Know the location and use of protective emergency equipments. This includes the eye wash station, safety shower, first aid box and fire extinguisher. 8. If scalpel blades, pins and knives are used, take care not to cut yourself. If you do cut yourself, call lab instructor immediately. 9. The hazardous chemical and volatile substance should be used in the fume hood. Thus, perform only experiments that your teacher instructs you to. 11. Always behave responsibly in the lab. 12. Always follow standard operating procedures, SOPs, and good laboratory practices. 13. Use holder to handle the heat objects. 14. Wash skin immediately and thoroughly if your body contacted with hazardous chemicals or microorganisms. 15. The laboratory working surface should be kept neat, clean and decontaminated at the end of the working day. 16. Ensure all Bunsen burners are turned off at the end of the practical session. 17. All used glassware must be thoroughly cleaned at the end of the practical session and all chemical containers should be properly sealed. 18. Avoid overcrowding and unnecessary talks with other lab workers. 19. Know how to dispose of lab waste properly. 20. Use first aid box if one gets injury from certain chemicals in lab. Personal protection. 1. Always wear personal protective equipment, laboratory coat, disposable gloves and safety glasses. 2. Avoid eating, drinking and mouth pipetting in the lab. 3. Do not use lab glassware to eat or drink. 4. Long hair, ties and scarves must be tied back. 5. Do not run around or play practical jokes in laboratory. 6. Wear open-toed footwear in laboratory. 7. Do not store food items in the laboratory refrigerator. 8. Never work alone in the laboratory. 9. Wash your hands with water and soap after removing gloves, before leaving the laboratory, and especially after handling a potentially hazardous material or microorganisms. Thus, in case of accidents or chemical spills, call your teacher, lab instructor immediately. 11. Any abrasions, cuts or open wounds in the skin should be covered with an adhesive plaster before beginning work. 12. Do not use cell phone while working in laboratory. 13. If you are pregnant or feeling uncomforted or sick, please let your lab instructor, teacher know immediately. First aid box is a dedicated kit to provide immediately treatment if one injured. It has the items like gloves, eyeglass, mask, bandages and pins, roller gauze, antiseptic wipes, sterile dressing, 2 triangular bandages, eye pad dressing, scissors and tweezers. First aid box should be easily accessible to all laboratory personnel. Gloves protect from hazardous chemicals, toxic reagents and infectious agents in lab. Fire extinguisher cylinder is used to control small fire in emergency. Laboratory workers were injured in 2019 due to explosion of chemical material at Forensic Science Laboratory, FSL, Bangalore, Karnataka, India. This chemical blast was occurred due to mishandling of chemical safety instructions. 1. You must be familiar with the potential hazards, chemical and gases. 2. Know how to use and handle equipments and chemicals before use them. 3. Notice all warning signs labeled on chemical bottles for safety purpose. 4. Many chemicals, solid, liquid or vapor, are poisonous, should be operated in the fume cupboard. 5. Never leave anything that is being heated or is visibly reacting unattended. 6. If a chemical is splashed into the eyes or skin, flush for 15 minutes. 7. Do not taste, mix and inhale chemicals for fun. 8. All reagents bottles should be labeled with the name of the chemical. 9. Always your face must be away from the mouth of test tube while heating a substance. Thus, be extremely careful when transferring, distilling or reflexing acid liquids. 11. Do not expose electric sparks, open flames and heating elements to organic solvent vapors which are flammable or combustible. 12. Never pipetting with your mouth. Mechanical pipette devices may be used for transferring all liquids. 13. Do not pour water into concentrated acid directly. Pour acid slowly into Water, stirring constantly near the walls of container. Mixing acid with water is often exothermic. 14. Pour liquids carefully into small container by using the beaker and funnel. When spillages occur, you need to call the lab in charge immediately to assist in cleaning up the spillage. Therefore, laboratory safety rules help us to keep safe and clean laboratory environment and protect all from exposure of hazards, chemicals and other solutions. The laboratory safety information, do's and don'ts, is summarized in Table 1.1. Table 1.1, Do's and Don'ts in Laboratory. Do's. Always wear safety glasses, lab coat and gloves for self-protection. Handle carefully of flammability, reactivity, toxic chemicals and reagents. Know the location of safety equipment. Proper disposal. Laboratory and work tables should be scrupulously cleaned with liquid detergents and disinfectants. Maintain lab notebook. Perform experiment as per instruction given by teacher or lab coordinator. Don'ts. Never work alone in the laboratory. Eating, drinking, smoking and storing foods in the laboratory. Unauthorized experiments, mouth pipetting. Paperwork should not be done on potentially contaminated surface. Idle gossip while working. Mixing chemicals for fun. The world worst disaster gas tragedy was occurred on the 2nd to the 3rd of December 1984 in India. Due to the leakage of highly toxic gas, methyl isocyanate, MIC, from the Union Carbide India Limited, UCIL, pesticide plant in Bhopal, Madhya Pradesh. About 5 lakhs people were exposed to MIC. People living around pesticide plant were died and badly injured due to the exposure of MIC. This incident is popularly known as Bhopal gas tragedy. 1.4.2 Laboratory safety symbol. Keeping yourself protected is the first priority of any student while working in laboratory. The common laboratory safety symbols are mentioned in Table 1.2. You should know what all of these means. Laboratory can cause a threat to the health and safety of workers or students at the workplace. These threats can be chemical hazards, physical hazards, biological hazards, H. Table 1.2, the common laboratory safety symbols. Look at the screen. 1.5 Summary. So far, you have learned that good laboratory practice describes good practices in organization. Process under which non-clinical lab studies are conducted safely for development and marketing of FDA-regulated products. GLP principles provides a framework under which studies planned, performed, monitored, recorded, reported and archived. GLP regulation gives assurance to regulatory authority that non-clinical lab data submitted are a true reflection of reliable, repeatable, auditable, and globally accepted. The data has reproducibility, integrity, accuracy, and consistency. OECD principle for GLP is intended to support and promote the universal quality management system and to ensure the safety of chemicals and chemicals product for human welfare and healthy environment. The fundamental points for GLP regulation include test facility management, quality assurance program, meeting the requirements of the test facility, equipment, receipt, handling, sampling and storage. Standard operating procedures, performance of the study, reporting of study results and storage and retention of records and materials. Laboratory is a dedicated place to carry out the experimental study. It supports science education in a real manner to test and validate the biological or scientific inquiry. To work safely in the laboratory, each student must follow the laboratory safety measures and rules. The common safety equipment are the safety goggles, eye wash stations, safety showers, lab coats, protective gloves, fire extinguishers, chemical fume hoods and first aid kits. Laboratory safety hazards symbol alert you about the safety of chemicals and instrument being used for experimental work. Having the knowledge of good laboratory practice and laboratory safety rules, you can prevent or reduce accidents and minimize damage that might occur in the laboratory. Always wear lab coat, goggles and gloves in the lab. No eating and drinking, gossip in the lab. Know how to use the laboratory equipment before use it. In case fire, know the emergency fire equipment like fire extinguisher cylinder is located. While heating a substance, mouth of test tube should be away from you and others. Use test tube holder while mixing or heating test tube over the burner. Flame. Do not mix or test chemicals for fun. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.